Okay, so for this second part of our lecture, we want to talk about uh, this area of gene regulation. So we want to continue with that discussion of gene regulation, right? And we are specifically talking about genes that are involved in development. And again, we're not going to talk about all of them because there's many, but we're going to talk about some examples and give you some examples of things that lead to issues with human development. Right. So first thing I'll mention is that there are things called homeoboxes and homeogenes, right? You probably heard about these, but these are clusters of genes that become active to cause the organs and tissues and things to develop as they're needed, right? Now, we're talking about them primarily right now in the idea of fetal development, where they must come on in very specific ways, but they're active throughout much of our lives, okay, doing various things. But anyway, a homeobox, as it's called, is where genes that are similar in form, very highly conserved, so they're formed, and they're in these domains, right? So what they do is that they're clustered usually on a chromosome or near each other's on various chromosomes. Although there are in humans, we're pretty sure right now there's about 235 of these homeobox genes. They're scattered all across all the chromosomes and forms. But again, once they're on a chromosome, as we'll talk about the Hox genes and things a little later, they're clustered close together, so they're like repeat areas. Okay. Now, once this gene is sequence is used, once it's transcribed and translated, okay, they produce these fairly small proteins that have units that have nuclear recognition areas, so they can be pulled back into the nucleus, and their job is really to be a transcriptional factor. Okay, so they're usually somewhere around 60 amino acids, and they contain areas that are specific for finding promoters and enhancers. What they tend to do is go in and lock on to an enhancer area or promoter area, so some upstream area from a gene, and cause the activation of that gene. In particular, there's the alpha-3 region, which usually expands the DNA and causes it to open up, so helps break those hydrogen bonds. Now, there's some other groups here. We're going to talk about the Hox group, but one of these gene families that's involved here are tail proteins. These are called atypical because they don't have exactly the 60 amino acid sequence. They don't look exactly like this, but they have a very important in that they're some of the first ones that often show up at the enhancers, and what they do is cause they're basically signaling for other transcription factors to come in. So they'll come in, they'll bind up to the DNA, they'll help to stabilize it, and then when it's stabilized and ready to go, that signals for other ones to come in. So these are homeobox genes, okay? They're just got to have a different names that we'll see in a moment. All right, so what's one good example, the classic example that's always used of genes that are homeoboxes are the Hox clusters. Okay, and Hox clusters, Hox genes are involved, as we'll see in a moment, in turning on in specific parts of the genome of the organism and causing development of an appendage such as an arm or leg, or there may be something that's involved in spinal formation, etc. Now, these genes have been around. They've been around for a long time. They're highly conserved, found in all sorts of areas. And as we move up, and they tend to be found in clusters. Okay, either clustered on a single chromosome, like you might see in the Cephalochordata, or they be spread all over the place. The Urochordata has them in a number of different of their chromosomes. Okay, but everything has them. Now, once we get up here, you see this says ray fin fishes, 7 clusters, and there were losses. Well, what happened when you get to the ray fin fishes? Okay, so it gets a teleost. There were these duplication events, so there were whole genome duplications. And once you get into the teleost, there were more duplications. So you basically had X number of chromosomes, and then you duplicated the whole number, and you duplicated the whole number. In fact, good friends of mine work on these fishes up in Michigan, and they're octoploids. Okay, they're big sturgeons, and they're octoploids. They're really hard to work with because they have 8 copies of every gene. And some of them have been deactivated and that sort of thing, but trying to understand them. Anyway, you've had these duplication events. Part of that duplication involved, of course, these Hox gene clusters, so that ultimately, by the time we get to humans, there's primarily 4 major clusters, okay, across all mammals and humans. And they're color-coded here because they have different functions that are related to parts of the body that they form, right? So let's take a look at those from humans, okay? As I said, we have 4 primary areas. We have one found on chromosome 7. There's a group found on chromosome 17, which is our group, 12, and then also on chromosome 2. Okay, so these are similar genes. They're gene families. Some areas they're basically repeated genes that are been modified. Other places they are fairly unique genes, but they're formed. So these are, you know, forms. Now this image over here and these colors coordinate. So as you can see that the Hoxa gene 1, 2, and 3 are involved in development of the head region, facial region, neural region, etc. The blue ones, which expand across all these groups, 9, 10, 11, and they're duplicates. And a lot of these are just duplicates, okay? So 9 is found not only on chromosome 7, but very similar ortholog found on 17 and 12, etc. The blue ones are involved in a major part of the formation of the body cavity, the mesothelium and all the internal parts there. And then the greens, which again, 13 is a version that's found repeated across the genome, as well as 12. These are highly involved in limb formation, okay? So making these limb buds function. And this is just an illustration of when you have fetal formation, where you see the activity of these, okay? The yellow ones being the formation, particularly in the vertebral column, and which ones are here. So HOX5 is active up here in the early parts, HOX6 is in the middle section, then HOX11 or 12 we see down here, and ultimately, again, those are going to be part of formation of buds. Okay, so what are these? Well, these are genes. They're controlled by other genes, right? Because they have transcription factors and things that come in and tell them. But once they turn on, they behave as transcription factors, and they move about the system actively. And that's how we get, you know, we need to have the spinal column formed, or we need to have eyes formed. Those stages that we saw, those Carnegie stages, these are being activated at various times during those Carnegie stages. Okay? Well, as you might guess, things can happen and go wrong, okay? So I thought I'd just give a few simple examples and show you, well, we do know that there are some situations and fairly well defined that relate back to these. One of those is we go to chromosome 7, so we're up here on chromosome 7, we're in the Hox region that's found in here. This is Hox gene A2. It has just 2 coding exons. Remember, these Hox genes are only fairly short, right? By the time they're mature, they're something like 160, because they're only going to code for about 60 amino acids in their form. So it's got 2 coding exons that are involved here. There are mutations that can occur that lead to a malformation of the ear, the external pinna of the ear, as well as malformations of the ear canal itself, okay? Sometimes leading to deafness in one side, or other times just you know, morphologically forms. It tends to be the predominant form, and there are multiple forms of this, okay? I think one of the recent studies found at least 40 genes that can be involved in non-syndromic mycotona. There's some very rare cases of others, but the fairly common form of it is an autosomal recessive, and it's due to a nonsense mutation. Okay, remember what nonsense mutations do, right? You have missense, which just changes the amino acids. You have silent, which don't do anything. You have a missense mutation, which changes the protein that's being formed in the sense that it changes the amino acids. And you have a nonsense, which are usually stop codons, right? And so this is a stop codon mutation that occurs in the homeo domain And because it's in the homeo domain, right, then that protein's not able to, that particular Hox form is not able to go and appropriately lock in where it needs to on the protein, on the gene that's involved in the production of these ears. So it gives you a form. And this is a little picture of the normal situation and signaling where it occurs and then which ones fail, okay? Another one is this idea of congenital facial paresis, and this version of it is due to malformation of the seventh cranial nerve. And so what it causes is that it causes the to not have proper innervation of the facial region. So you get a droopy face, okay? And there's been there's again, a number of forms of this. But at least one of the common forms that we see is an autosomal recessive as well. That's been determined by sequencing some families where they do into the pedigrees, right? This is a pedigree over here. And several of the children, it was a large family. And so these individuals, remember I told you that some pedigree symbols, when you know it's a carrier, you have a recessive, you'll use a little dot in there. So both mother and female, mother and father were carriers. So they're indicated with little dots inside of their symbols. We see the children are born, and this is reverse birth order. They had children 1, 2, and 3, and then they had 4, 5, 6, 7, 8. So it's flipped, but that's okay. First 3 children all have a mutation. So if we look at mom and dad down here, they were heterozygote carriers for the CT mutation. Okay? And each one of these children got a T. Okay? And that made it into a recessive condition for them. Okay, and it had to be, now think about what has to happen for that T to be the only thing there, for these not to be heterozygous children. Mom had to give a T, so they had to give the mutation. Dad had to transmit the T, which is also a mutation, to make them a homozygous recessive. Okay, all these other children have a large family. All these other children are either heterozygotes or they could have gotten C. Okay, there's some other forms. So it's classic Mendelian genetics going on here. This thing's a form. And what it's doing here again is the Hox ONB gene is found on chromosome 17. It is once again messing with the primary domain that we've got here. So it's a replacement of the arginine at position 230, and we see that we don't get good lockdown again. Okay, and in doing that, you end up with this seventh cranial nerve not forming correctly. Okay, not being fully mature. Cranial nerve coming forward this way, and in the facial droop, the eyes lids aren't able to function completely correctly, and you just get a full facial droop that's found here. So it's a Hox gene problem. Okay, there are again a number of different variants of this, but this is one of them. Okay? And then the last one I want to say is not a Hox, but it illustrates how other developmental systems and how complicated those can be. Okay, cinepondetti is when is a combination of polydacty. Polydacty is when you have an extra digit, or it can be post-axal polydacty, so it can be somewhere after the knuckle, or it can be of course on toe, wherever, but you end up with an extra part of your digit. Okay, this particular system actually has a combination of that. There are, as you can see here from these x-rays, there's 1, 2, 3, there's extra digit parts in here, okay? Or here's post-axal, but then once they're there, they fuse, okay? So instead of having 1, 2, 3, 4, 5 separate digits as we might have, there's actually a combinational digit in here. So you end up with individuals that either have too many, here's an extra one here, okay? Combined with the fact that you have a fusion going on to give you this form. Now, these are also dealing with a homeobox, right? But it's a different gene system. Okay. Here we're talking about a gene system found on chromosome 2. This is the Hox D13 system, and we are seeing that you get this sort of whole series of mutational problems. Okay, here's the number of mutations. Okay, or the location of the mutation as they're calling it. Here's the type of mutation that you have. Okay, And some of them are nonsense, some of them are expansions, there's frame shift mutations, there are all of these different ones that can lead to this relationship between this homeobox, okay? Now this one is an autosomal dominant, so everybody that's gonna, everybody that gets one copy, even a het, expresses this double problem that we see here. Okay, so again, this is yet another Hox. It's a different Hox form than we saw in the last ones, and there are a whole bunch of problems that can arise depending on where the mutation occurs. And each one of these little things shows you some of the places. Here's a location 1 that's non-specific. Let's see which one. Here's one that's a frame shift is at 14. So you can either be an exon 1 or an exon 2, part of the homeobox or not part of the homeobox. Okay. All right, so those are all ones that are associated with Hox systems, okay? And the last one is not Hox system, okay? As I said, I wanted to show you one that wasn't after that last one because I concentrated on Hox, but there are other types of homeoboxes, okay? And the PITX1 gene found on chromosome 5 it's involved in signaling through the pituitary, okay? It's its ultimate job. Okay, it's a transcription factor, but it moves over there. It's also an autosomal dominant if you have a mutation in the form. And so what you see from this form is that the growth of both the hind limbs or whatever can be impacted by not having the expression of these or having overexpression of these. In humans, okay, we have a leitenberg syndrome, like Leitenberg syndrome, which is an autosomal dominant. And it is the malformation of the limbs in many ways. Okay, essentially, you don't get the full formation of the elbows or the wrist or whatever, and you get these locked systems, which are just pretty much inflexible systems. Okay? And it often occurs. It can occur in the knees, it can occur in the ankles, but most certainly for humans, this syndrome is particularly found in the arms and hands. Okay, now there are 2 causes. Right, there are causes. One of them, as it says here, is seen as a series of base mutations, 180 to 269 base mutation. That it occurs though in this region right here. Here's we got the genes here. Okay, the base mutation is not actually in the gene itself. The mutation occurs in the area between the gene and its enhancer. This 1473 is the enhancer region for it. What it does, it actually moves the enhancer closer to the gene itself. In doing so, okay, it doesn't move quite that far, but it moves it closer. And in doing so, what it does is it causes overactivation of the gene. So this particular problem is not caused by failure of the protein to be produced, okay? It's actually, as you can see here, this is one where you would have a sort of a normal production. You have overexpression. This is overexpression of the gene itself. This would probably be in the middle here where you expect to see some type of normal expression. And this is where you can see the protein's been marked in the fetus and you get overexpression, okay? There's a second version which involves a translocation between chromosome 5, where this is found, and chromosome 18, okay? And what this translocation does, remember translocations are where you take one chromosome and you combine it with another one in some break in some way, and either you may gain DNA on one chromosome or may lose it, okay? You have translocation carriers. We've talked about the effect you have or will in earlier genetics courses when you talk about things like familial Down syndrome, right? Where you have 1421 translocation, where most of the 21st is stuck on the 14, and those people that are carriers look normal, but they produce children that are monosomies or trisomies, etc. Okay. Well, here's one where you mess it up. Well, again, what happens here is that you once again have enhancers put in the wrong place. You're bringing enhancer pieces off of chromosome 18, putting them here, and they hyperactivate this particular gene. And this is an example over here of a fetus that's been stained to show the hyperactivity of that gene. So in this case, we've got a Hox, excuse me, we have a homeobox gene. There's a series of genes here that are involved. Okay. We have not a mutation like we did before, right? Before we had mutations specifically, many cases in the homeobox or in the actual gene itself, right? Because this is in the Hox gene here. But we have something where we have the regulatory region being impacted in some way, either and not necessarily by mutation of the regulatory region itself, but by the location of the regulatory region. So we get all sorts of different forms. So what's all this? Well, I'm just trying to show you that if in development, you have lots of genes involved. You have a whole series of things that can occur and not just one thing. So when people go out and they try to figure out, okay, what's going on in this particular case right here? Well, it can be, dozens of things, and in fact, hundreds of things, right? Very often, and as we're doing more human sequencing, things that we have thought of, okay, it's an autosomal dominant, got that, right? Both of these versions are autosomal dominants. However, you can also have very similar outcomes from timing problems, from other genes not activating, because obviously there's a multiple pathway here, just like there was in sex determination or anything else. To make a limb and to get those fingers and the bones and all the cartilage replaced so that you get bones and all that, you end up with all sorts of possibilities that can occur. Well, that basically concludes what I wanted to say about transcription at this point. Excuse me, about the development at this point. We'll bring more in as we talk about some of the other, when we get to more syndromes, we'll highlight the fact that there's a Hox gene involved, or that there's a secondary effect that leads to developmental, particularly in things when we talk about neural systems. But this is, I think that should cover just a broad overview of some of the developmental issues that relate to human genes and how human genes in the human systems play a role in development.